In this video, we are going to look at the transport of carbon dioxide. So, we have already discussed about the transport of oxygen and we have completed it. So, now in the exchange of gases, the second type of gas that the blood transport is the carbon dioxide. So, now let's look at how the carbon dioxide is transported in blood. So, we have learned that carbon dioxide in the blood is transported in three forms. One is in physical solution as carbonic acid, that is about 5% of carbon dioxide is transported in the form of carbonic acid dissolved in the blood plasma. Now, the second form is as hemoglobin compound, that is carbaminohemoglobin. About 10% of carbon dioxide is transported as a compound or combined with hemoglobin as carbaminohemoglobin. And now majority of carbon dioxide in blood is transported in bicarbonate as bicarbonate. So 85% of the carbon dioxide in blood is transported as bicarbonate. So now let's look into each of the methods in detail. So the first form, that is carbon dioxide, is transported in the form of physical solution as carbonic acid. So carbonic acid is formed by combination of carbon dioxide and water. So the carbon dioxide which is formed in tissues, it is going to diffuse freely into the plasma. From tissues, the carbon dioxide is going to diffuse freely into the plasma, and there it is going to combine with water and forms carbonic acid. So in the plasma, there is abundant water, and in the plasma, it combines with water and forms carbonic acid, that is H2CO3. So H2CO3 readily diffuses or dissociate into HCO3 minus, that is bicarbonate ion, and H plus ion. So this step is catalyzed by an enzyme called carbonic anhydrase. So in vertebrates, the carbonic anhydrase is present in the RBC, while in case of invertebrates, the carbonic anhydrase enzyme is present in the plasma. So in case of vertebrates, only a very low amount of carbonic acid is formed within the plasma, since the enzyme for catalyzing this particular step or this particular reaction is absent in the plasma. So it is the formation of carbonic acid is very fast in the case of invertebrates, and while in the case of vertebrate, formation of carbonic acid within the plasma is a very slow process due to the absence of carbonic anhydrase enzyme. So that's all about the first mode of transport as in the form of carbonic acid. Now we'll move on to the second form of transport, that is in the form of carbaminohemoglobin. So also the carbon dioxide diffuses into the RBC. So there inside the RBC you have the hemoglobin. Hemoglobin is the red pigment that helps us in transporting the gases, both carbon dioxide as well as oxygen. So we know that hemoglobin has got two parts. Hemoglobin has a heme part as well as a globin part. So it has a heme part and globin part. Here the globin part is a protein part. So globin will have abundant amino groups, that is NH2. The amino groups are obtained from the globin part, and the carbon dioxide is going to combine with those amino groups. So here you can see the combination of carbon dioxide and the amino groups from hemoglobin. The amino group from hemoglobin is designated as HbNH2. So in the tissues, they combine together and form this particular compound. This compound is known as carbaminohemoglobin. Carbaminohemoglobin. So in tissues, the carbon dioxide readily combine with the amino group from the globin part and form carbaminohemoglobin. And the carbon dioxide is transported as carbaminohemoglobin through the blood, and only 10% of the carbon dioxide is transported in this manner. Now, when the carbaminohemoglobin reaches the lungs, what happens is the reverse of this reaction is going to take place. That is, carbaminohemoglobin easily dissociate into free carbon dioxide, and the amino groups are now free. So the the free form of carbon dioxide which has dissociated from the carbaminohemoglobin is going to enter the lungs. So the lungs will be filled with carbon dioxide which has now dissociated from the carbaminohemoglobin. So now the carbon dioxide buildups, and what happens is it is going to be expelled out through the exhaled air. So that is how carbon dioxide is transported in the form of carbaminohemoglobin. Now we are moving on to the third method of transport, that is in the form of bicarbonates. And you have to note that 85% of carbon dioxide, that is majority of carbon dioxide obtained from the tissues, it is transported in the form of bicarbonate. Now we'll see each in detail. So in order to see the process in detail, we have to depict two things here. So this thing is the tissue, and this I've shown here, that is the RBC. This is the RBC, and here you can see the space between tissue and RBC, that is the plasma. So the RBCs are suspended in the plasma, we know that. So what happens is, as a result of metabolism, metabolic waste product, that is carbon dioxide, it is going to accumulate in the tissue. So here carbon dioxide is accumulated as a result of metabolic activities. So now what happens is, carbon dioxide diffuses first into the plasma. So it is going to diffuse into plasma. So now it has reached in the plasma, and from the plasma it readily diffuses into the RBC, inside the RBC. And as soon as the carbon dioxide diffuses into the RBC, now it is here. Okay, so now our carbon dioxide has reached inside the RBC. So now let's stop it there. Now let's see what are the situations in the plasma. In the plasma, we know that there are different kinds of salts. So one of the salts present in the plasma is NaCl, that is sodium chloride. So sodium chloride dissociates in the plasma and give rise to sodium ion, that is Na plus, and next is Cl minus ion. Okay, so this NaCl present in the plasma is in the form of sodium ion and chloride ion. So now let's go back to the carbon dioxide which is waiting in the RBC. So once the carbon dioxide gets inside the RBC, it is going to combine with the water inside the RBC. And what is the result of combination of carbon dioxide with water? It gives rise to carbonic acid, that is H2CO3. So this reaction, we have seen this reaction earlier, and the enzyme that catalyzes this reaction is the same carbonic anhydrase. And remember what I've told you, the carbonic anhydrase is present in high quantities within RBC in the case of vertebrates. So this reaction, that is conversion or the formation of carbonic acid within the RBC, is very fast due to presence of carbonic anhydrase enzyme within the RBC. So as soon as the bicarbonate is formed inside the RBC, it is going to readily diffuse into two. Which are they? That is bicarbonate ion, that is HCO3 minus and H plus. So these are the two dissociation products of carbonic acid. Now what happens is, the H plus formed inside the RBC, the H plus cannot stay as a proton, that is H plus. The hydrogen ion cannot stay as such. So what happens is, the RBC now have free hemoglobins. So the blood was carrying oxygen towards the tissue. It has unloaded its oxygen, so the hemoglobin is now free within the RBC. So such hemoglobin that has already delivered the oxygen it had carried, that hemoglobin is known as a deoxyhemoglobin, a hemoglobin that has just delivered oxygen, that is free of oxygen. Such hemoglobin is known as deoxyhemoglobin. So what happens is, this deoxyhemoglobin now it is going to act as a proton acceptor. So deoxyhemoglobin is now going to act as a proton acceptor, and it is going to accept this H plus formed as, or this H plus which has dissociated out from the carbonic acid. So now these two combine and form a product known as hemoglobinic acid, HHB. This product is known as hemoglobinic acid. Hemoglobinic acid. So the formation of hemoglobinic acid is known as a particular effect that is known as Haldane effect. So what is Haldane effect? The deoxyhemoglobin accepts the proton which has formed as a result of dissociation of carbonic acid, and this results in the formation of an acid called hemoglobinic acid. This effect or this phenomenon of formation of hemoglobinic acid is known as Haldane effect. This effect is named after a scientist whose name is Haldane. So that is why it's known as Haldane effect. Okay, so now H plus is being taken care of. Now what remains is the bicarbonate ion which has dissociated out from the carbonic acid. So now what happens is, if the bicarbonate ions are going to build up in the RBC, obviously it is going to build up. How? Because the tissues are always producing carbon dioxide. It is going to readily diffuse into the plasma, and then it is going to diffuse into the RBC. So there is going to be a buildup of carbonic acid, and as soon as carbonic acid is formed, it is going to diffuse into the bicarbonate ion and H plus. So the buildup of bicarbonate inside the RBC is not good. So it is going to exit the RBC, or it is going to move out of the RBC as soon as it is formed. So what happens is, this bicarbonate which has dissociated from the carbonic acid, it is going to slowly move outside of the RBC into the plasma. So now the bicarbonate ion has reached inside the plasma. So now we know that what is charge of bicarbonate ion? It is a negatively charged ion, HCO3 minus. So a negatively charged ion has moved or exited the RBC. So that results in an electrical imbalance. So a negative charge has moved out of the RBC. That results in an electrical imbalance within the RBC. So in order to compensate for the loss of this one particular ion, that is one negatively charged ion is lost or moved out of the RBC. So in order to balance or compensate this loss of negative charge, another negative ion from the plasma, it is going to migrate into the RBC. That's how the loss of a negative charge is compensated. Simply, another negative ion is going to move into the RBC. That's how the electrical imbalance is now compensated. So which ion is present in the plasma that is negatively charged? So I've already mentioned about the dissociation of NaCl. So we have already seen that NaCl dissociates into Na plus and Cl minus. So how Cl minus is present? It is a negatively charged ion. It is present in the plasma. So this Cl minus ion is going to shift or move into the RBC in order to compensate for the loss of a negative ion, that was bicarbonate ion. So this movement of negatively charged chloride ion, in order to compensate for the electrical imbalance of a negatively charged ion within the RBC, that movement of chloride ion is termed as chloride shift. Chloride shift. Why? Because chloride ion has shifted from the plasma into the RBC in order to compensate for the loss of a negatively charged ion, that is bicarbonate ion. So this chloride shift, this phenomenon is also known as Hamburger's phenomenon. Hamburger's phenomenon or Hamburger's effect or effect. Okay, so now the chloride ion has reached into the RBC as a compensation for the loss of a negatively charged ion. So now what happens is the chloride ion, once it has reached inside the RBC, it is going to combine with another positively charged ion which is already present inside the RBC. That is nothing but K plus ion, that is potassium ion. So once the chloride has reached inside the RBC, it is going to combine with a positively charged ion, that is K plus ion, and forms KCl, potassium chloride. So it has formed KCl. Okay, so that is how chloride is going to exist inside the RBC. That is by combining with potassium and forming potassium chloride. So the chloride ion is being taken care of. Now let's talk about the bicarbonate ion that has migrated outside of the RBC or into the plasma. So here is a bicarbonate ion. So what happens is once the bicarbonate ion has moved out and reached the plasma, it is going to combine with another positively charged ion. So, which is the positively charged ion which is present here? That is Na plus sodium ion. So, it is going to combine with the sodium ion and form sodium bicarbonate. So, bicarbonate on combining with sodium ion, it is going to form NaHCO3. That is sodium bicarbonate. So, the sodium bicarbonate is formed within the plasma. So, this sodium bicarbonate is carried to the lungs by the blood. So, this bicarbonate is transported inside the blood or within the blood to the lungs. So, once the blood reach the lungs, what happens is the NaHCO3, that is sodium bicarbonate, it is going to dissociate into sodium and bicarbonate, and from the bicarbonate carbon dioxide is going to be liberated, or the carbon dioxide dissociates out from the bicarbonate ion, and all the processes that has taken place here, all the chemical process that has taken place here, that is going to reverse once the blood reach lungs. Okay, so now carbon dioxide has dissociated from the bicarbonate of the sodium bicarbonate, and now that carbon dioxide, the free carbon dioxide, now diffuses into the lungs. And now the lungs will be filled with carbon dioxide carried by the blood in the form of sodium bicarbonate. And once the lungs is filled with carbon dioxide, it is going to expel the carbon dioxide during exhalation process. So that's how we are going to remove carbon dioxide by accepting it from the tissues, transporting it in the form of bicarbonate, and uh, you know reaching or unloading the carbon dioxide within the lungs. So, in this mode of transport as bicarbonate, we have to focus on two effects. That are, one is the Haldane effect, and next is the chloride shift. Chloride shift is very important, and you have to provide this particular diagram while explaining the chloride shift or Hamburger's phenomenon or Hamburger effect.